So it's my great pleasure to introduce Dr. Viktor Jirsa. Dr. Viktor Jirsa is the director of the INSERM Institute de Neurosciences des Systèmes at l'Université Aix-Marseille in Marseille, in France. And he received his PhD in 1996 in theoretical physics and applied mathematics, and has since then contributed to the field of theoretical neuroscience, in particular through the development of large-scale brain network models based on realistic connectivity. So with Viktor, I met Viktor in 2001 for the first time. And this is at the information processing in medical imaging in Davis in US.
Davis, this was...
UC Davis.
I forgot about that.
Oh, yes. This is my the opportunity to say 2001.
Yeah, but the name, the conference, it was
IPME.
IPME, exactly.
IPME workshop. And I remember that he got the prize of the conference at that time. So we become a little bit famous just afterward. But it was really, and his contribution was in neural field, in EEG and MEG. So he has since then really a big and a huge contribution in our domain in neuroscience. Just to let you know that it started before, I think, in Florida when you did your PhD, but at the international level, I see him for the first time in 2001. So this is like the history of les anciens combattants. This is what we say in France. So his work has been foundational of network neuroscience in brain medicine and how to use personalized brain models in epilepsy. He is the scientific director of the clinical trial EPINOV, evaluating the use of virtual brain technology that you heard about it yesterday with Dr. Randy McIntosh in epileptic surgery. Dr. Jirsa served as a chief official in the European Digital Neuroscience Infrastructures, EBRAINS, and lead investigator in the Human Brain Project. You know, how big is this infrastructures at the European level? So Dr. Jirsa has been awarded several international prizes. As I mentioned, I think that the first prize was IPME, but I'm not sure. So for his research, including the first HBP Innovation Prize, and Innovation Prize was in 2021, and the Grand Prix de Recherche en Provence in 2018, and has published more than 160 papers and probably more. So it's my great pleasure having you with us today. And okay, thank you for accepting our invitation. The floor is yours.
Thank you very much for this very kind introduction. You touched me, actually. Huh? Hit me, yeah. What I presented at IPME. So 2001 it was, you say, yeah? There was some very early work that was, Randy, before I even met you, that was the beginnings of the neural field theory, linking it to forward solutions and actually bridging the gap between neural signals and neural modeling. But at that time, connectome, you remember when the first connectome paper came out? It was about 2005. Yeah? The word connectome did not exist at that time. And one of what we showed at that time, only naive approximations of connectivity were done, typically with a high symmetry. And we have demonstrated that in order to ever bridge the gap to realistic signals, you need to take realistic connectivity into account. And at that time, all diffusion tensor imaging, all of this was upcoming and became a reality. So we actually said we need to integrate it. And that was a little bit the very modest beginnings of what we call the virtual brain. And then I ran into Randy and it became reality. Okay, we have switched the order. What I will do today is I will give you a high-level overview of some of the work. that we have done, particularly in epilepsy, so applications in brain medicine, but also an insight of the functioning of EBRAINS. Not too much, but enough to make you go to the website probably and have a look at this. And by the way, I also brought you, not a present, but there is a tool book that came out of this. All the softwares that I will talk about that are available, yeah, on the EBRAINS account, and I invite you to have a look at this. Maybe you can pass it through and people can have a look at this, yeah? Just hot off the press. So, and in the second lecture, we will do a little deep dive into some of the intricacies, what works, what doesn't work, so more technically. Yeah. So, Sabib mentioned much of the work that I will speak about comes out of the Human Brain Project and actually precedes the Human Brain Project, as we just discussed. The virtual brain, for instance, was released as a platform in 2012. The Human Brain Project started around 2013-14 and finished last week. Yeah. So this is 10 years later and a big sum of money, about 600 million euros later. This is where we stand. And the transition is occurring into EBRAINS, which is today a European infrastructure, so not a project, infrastructure, which means it doesn't have a definite end, and is supported by the European Commission. And it provides us with an ecosystem for digital neuroscience, helping us in principle to study fundamental mechanisms of brain function and translation into clinics and technology. So one of the big elements that came out of the Human Brain Project is the digital twin approach. What does that mean actually? When you look it up, Wikipedia or something like that, you will find out that this terminology is actually fairly recent, 2006. Yeah, goes back to NASA, yeah, when they set up the first digital twins in industrial applications and manufacturing, building a cycle of fusing experimental data with modeling data in the same framework, operating it, performing, trying to individualize it specific to the particular platform or flying object they were looking at, and then use it in autonomous simulations in order to feed back information. Yeah? And this is what we are trying to do in EBRAINS, to provide you with the type of technologies that are necessary for that. So it's a cycle. Yeah, don't think of a digital twin as naively as as good as possible copy of the biological system. No, it goes way beyond that, and I will actually exploit this here with you. Yeah? So essentially, the approach today is show you with some principles of how to build a digital twin brain. Randy showed you already some of those yesterday. I will do the same with a different perspective, but same story, and then go into more depth in epilepsy. Yeah? So first of all, in terms of building it, what we want to do, in particular also EBRAINS, to support that, providing a full modeling platform on the brain level, but also for other structures, such as the microcircuit level, as you see represented here, and all the way down on the cellular level. So what you will find on EBRAINS are different representations of macro, meso, and micro scale. And what you will not find yet, but this is something we're working on, we want to provide you with standard models, essentially off the shelf, that you then can adapt to your needs. Don't think that this adaptation process is easy. It's actually one of the biggest difficulties that you will have, that we will have, in order to make it adapted to the scientific questions that you want to ask, either mechanistically or if you want to adapt it to your patient. So it will require some support, some computational support, yeah, in terms of storage, computation, HPC, so high performance computing, but also high throughput computation. And this is initially the Human Brain Project. Initially, we thought the majority will be HPC, high performance computing. We were wrong. Yeah, the majority is actually high throughput computation, Sometimes it goes under the label also under cloud computing. Yeah. So this we had to make true, and it is essentially a support that needs to be adapted to the workflow that you want to have, parametrization. Yeah. So I stay on the cartoon level at the moment, but I want to get you familiarized with some of the elements that go in there. So this is a parametrization of the models, but what we want to do, we want to integrate it together with different sets of data. The connectomics we discussed yesterday, which did not exist in 2001, but things came up. Multi-scale, very important. Yeah. So multi-scale representation of data going from the centimeter to the micrometer scale. And this needs to be kind of integrated, but or cohort level if you want to touch cohort data. Now you have to put this together with in vivo data. If you want to perform clinical translation, you need to specify it to the individual patient. Yeah. So you have actually the challenge to make ex vivo, in vivo, and ex vivo work together. Yeah. And our task was then to make workflows out of this, make these things work together, put them into an integrated framework. And if possible, and this is what this is supposed to show, a single entry, multi-user framework allowing clinical applications, surgical applications, applications in robotics or medication. Yeah? These things exist today, yeah, and some of them I wish to show you. So this is a framework, and that was the task, all that's supported by the underlying infrastructure. And this is what EBRAINS does. What we want to do in EBRAINS is to make sure that you are not bothered by this, but knowing where are the data stored, how do the tools interoperate, etc. Yeah? So this has then given rise to the science workflows have guided us to develop appropriate technical workflows that we then built around 5 pillars. And this is what you find in EBRAINS today. Atlases, data and knowledge needs to be organized following certain FAIR principles, simulations, brain-inspired technologies, and medical data analytics. And these are essentially, they themselves built 5 network nodes that we needed to make interoperable. And that was actually our task, supported by a hardware that is allowing you to seamlessly access compute resources, store data, be legally compliant with European data protection laws, things like this that has become very difficult over the past 5 years due to GDPR. So handling data following FAIR principles, organizing data in atlases, multi-scale atlases, simulation on multiple scales. If possible, they have to speak with each other, and derive brain-inspired technologies and build environments in which you can actually operate data as a clinician, your own data, or if possible, with appropriate data protection laws, working with other colleagues. Yeah? It's supported by European member states. 8 member states are full members today, together with quite a number of large institutions that are providing also in-kind services, in particular the 5 supercomputing centers in 5 member states providing HPC services, cloud services, and the appropriate data storage that is necessary for that. All this for everyone on this planet with an academic account, it is free. Yeah. And you as a clinician, you know how expensive it is to store data. So we get more and more clinical researchers actually turning towards us because the costs are actually skyrocketing. And within the reasonable limits, this is essentially what EBRAINS is providing. Yeah? So what I want to do now, this is the framework. I want to jump into more depth in the organization thereof and show you how we actually connect the tools, the models, and the data in order to enable this flow that I was just in a cartoonesque fashion describing to you. So the key anchor of all of this is the atlas. Yeah. So in the atlas, you represent data on different levels, region level. And it's not only one instantiation, you have to have a representation of the variability of the data also, because this is one piece of information that we use quite a lot. And at across multiple scales, yeah. So a region, on the region level, we are on the centimeter, millimeter level, and we can go down all the way to the microstructural detail, having representation of cytomaps, cytoarchitectures. There we are on the micrometer level, where data are coming, for instance, from polarized light imaging. I will show you on the next slide an example. And this needs to be organized also across modalities, yeah, functional modalities, structural modalities, ranging from the cellular level all the way to the connectomics level, and with the temporal domain, the functional data. Yeah? And that allows us then to, in this reference space, to go and put in your PET data, for instance, that are coming from your individual scanner, map it on posterior probability distributions, or here in this case, cortical depth, and this can be mapped by an ontology on the parameters in the models. Yeah? So this is not automatized, but it's enabled and interoperable. So you can actually, it's called Zebra. This has been developed by Timo Dickscheid. It's a tool you can navigate in this space, zoom into a particular region, open a pull down menu, and there you have all the different possibilities that you can visualize. This is actually coming from one of the visualizations. Yeah. So this is a form of operating. Like Randy, like us, we did in the virtual brain, we provide a graphical user interface, and this is nice. After you worked with it for 2 weeks, you want to go faster, more automated, etc. And everyone is jumping onto Jupyter Python notebooks. And this is a preferred means of use. Yeah, but it's a beautiful entry point to go through the GUIs. Yeah? And the models that we use for the virtual brain is essentially regional representations. And there we use mean field models. There's not much that I need to say about this today. We have spoken about this yesterday, but it's essentially mean field models are stochastic reductions of detailed single cell population models that are using statistical tools in order to obtain reductions of the inhibitory and excitatory neurons, and typically you obtain a reduced dynamic systems. Janssen-Ritt was mentioned yesterday. I think we have about 20 models in the virtual brain available. So the structure, this is also very powerful, is agnostic to the model that you want to use. Yeah, the mathematics that you obtain has this particular structure. Later in the second lecture, we will talk more about this. Psi dot x being the location. This is your internal dynamics at the network node. This is nearest neighbor connectivity, intracortical. Decaying, translationally invariant, which has certain mathematical properties, namely diffusive properties. Yeah? And this is a connector. Yeah. And about this, I spoke at IPME in 2001 already, pointing out that this is not translationally invariant, and we approximated by that. And that never was possible to fit experimental data to represent them correctly. We needed the symmetry breaking in here in order to move forward. And this is what became the connectome-based approach. Yeah? And so let me summarize this. We reconstruct from imaging data of an individual the cortical and subcortical surfaces. We parcellate it. Then each region we represent by neural mass. We connect it by the connectome. This I'm quoting the mathematical equation that I just showed you. Yeah, and then in addition to this, we have it spanned in three-dimensional physical space. So this is on the source level. We can stick in sensors, such as an EEG, so-called stereotactic EEG, to measure sensor activity, the way that the clinician or researcher is measuring it. So we are not limited to the source level in terms of modeling, but we can actually mimic the patient's own implantation. Yeah? And EEG, MEG, functional MRI. Yeah? So this is approximately the situation. So this video yesterday briefly. This is a simulation. Once you fix the parameters, there are no changing parameters. You make the simulations on the source level, but you project on the sensor level, and this is a time series that you see. And key to this, that you're capable of doing this, is that you have a representation of the fast discharges, but also the slower processes that can be captured. For instance, you spoke about astrocytes and glia, yeah, or extracellular effects that are linked to this, yeah, such as extracellular potassium. Yeah? So this is the environment. This is what the data look like when you measure it from the patient. Yeah? This is work from Cathy Chavon's lab. In the US, there's an electrocortical, so the grids are placed on top of the surface of the brain. This is the representation that you see here. There where the red cross is, she placed a microelectrode array, yeah, where you can actually, on the reduced scale, sub-millimeter scale, you see actually the organization of the waves that you see evolving here, that you see actually on a very slow scale evolving there. This is a particular patient, and there is a very rich dynamics during the seizure of propagating waves, spiral waves, very classic spatiotemporal structures. So this is actually the challenge that we have that we need to capture. And this gets us actually to the first next step, which is actually we need to jump up a scale on the space. What Randy showed you yesterday, with a connectomics-based approach, you have typically 150 to 250 brain region. This gives you this size of a brain region which resembles something like that, yeah? 16 square centimeters. This is a little more, but think of half of it. 16 square centimeters is huge, yeah? So what we have done over the past 3-4 years, we made it possible to jump up by a factor of 1,000. Yeah? And that gets us into the millimeter range. And this is not a triviality. It's not just computational resources. There are many things that change. On the neuroscience level, yeah, you're getting on a level where you have to take intracortical connectivity into account. You cannot ignore this anymore. U fibers become relevant. You need high resolution data to inform this. The bottleneck is not the simulation. The bottleneck is actually the model inversion, trying to make the estimation and inference that I have not spoken about. There are so many bottlenecks that go beyond just adding a few vertices, well, factor 1,000 more vertices, but it changes also the mathematical formulation. You're now in a field theory, not in a discretely coupled differential equation, even though it's functional differential equation situation. Yeah. So this has changed a lot, and it has become only possible over the past few years to work with that. Yeah. So I'm going to give you a few examples thereof. It has accelerated us enormously, in particular also for the situation with regards to the source to sensor mapping, which becomes important when you want to take it very seriously that you're performing personalized brain modeling. Yeah, you put lots of effort into putting sensors in there, and then your brain region is that large. There is no proper implementation of the physics of the electromagnetic theory mapping source activity to sensor activity. On this level here, it's becoming possible. Yeah? So here you see a representation of the connectome. This is now the level at which we are. Left hemisphere, right hemisphere, interhemispheric connections. So please note here we are on the level of over 100,000. Subcortical areas, subcortical areas, cerebellum. Yeah. So on how to make this is we built initially a first grid with this with a resolution of multiple square millimeters. We built the vertices of the surface, and then we extract the streamlines and we compute their intersection essentially with each vertex, and this becomes then the connection point, allowing us to build the high resolution connectomics. Resolution in terms of connectivity, we're on the 200 micrometer level. Yeah, this is ex vivo. Yeah, we need to connect it in vivo with data from the individual patient. I will show later how we're going to do this. Yeah? This is the type of data when you connect yourself, you find available to your use, and we are using it also for building high resolution templates. This is coming from Jülich, polarized light imaging. They take a brain and they have scanned it, yeah, in a particular preparation. They have scanned it multiple times and then sliced it, yeah, and used polarized light to actually. There are different techniques. Yeah, you can either penetrate it and refract it, or you can just measure the polarization, etc. It gives you different images. This here is PLI, and from the polarization, this gives you the angle of the orientation of the fibers. Yeah? Here we are on the resolution of 200 micrometers, and look at these beautiful pictures with the resolution of the human brain. Yeah? This is what we have available and represented in this brain reference space where you can use it actually as a template. Then from this allows us now that we have a structural representation thereof to build more detailed representations of areas that were not accessible before. This is a representation of the hippocampus. Hippocampus is actually part, when you go from the cortex, it folds very nicely into this structure here. We can actually unfold it, inflate it and unfold it and map it on a sphere, and that allows us then to do things like this, yeah, where we go directly from the cortex into this structure. Here you see the individual regions that I represent here now on the sphere. On each vertex point and neural mass represented with intracortical connectivity and cortical connectivity, and increase the excitability such that at the CA1 region, you start the emergence of this type of propagating waves, the way we have seen it from Chevon's data and that we find in animal experiments. This is a classic scenario you see. You see the propagation going to the frontier, and that is then traveling forward. It starts slowing down. This is a spike wave complex towards the end of the seizure, and then it stops. Yeah? So on this level of resolution, this is becoming possible today. So far, this CA1 region was just 1 point. Yes.
Your atlas is based on how many subjects?
One subject.
Only one subject?
It's one subject. It depends on this atlas, the way the big brain is based on one subject.
Yeah.
The Chenonceau is also one subject, but a different subject. What we can do if we want to make it patient specific and do not go to these very high resolutions, then we have to stay on the resolution of 1 millimeter, and this is what we do all the time.
Yeah.
It makes sense to go to this very high resolution if you have mechanistic questions, for instance, about epileptogenesis. We are not asking questions about epileptogenesis, sprouting and reorganization of the mechanisms supporting epilepsy. We are asking questions about ictogenesis, as you have seen here, and the ictal propagations.
Mm-hmm.
There, typically for us, millimeter resolution is sufficient. Yeah? And this we can do for subjects that do not have too much atrophy in their data. Yeah, and then we can make this type of reconstructions. Yeah? You have another example. And this is now for one subject. This is not this very high resolution. This is the subject's own data, reconstruction, rhinal cortex, the fibers coming from the rhinal cortex within the hemisphere projecting on the other one, representation of the connectivity strength. And we increase the epileptogenic zone value. And when we do this, please have a look at this. The seizure will start here. Theoretically, look at this point here. You see how the activity came up. It did not propagate through the cortical sheath. This came from the interactions. This is not possible outside of a system where you do not have the heterogeneous connectivity through the white matter fiber system. Yeah. So these are the type of symmetries we were talking about. It's simply impossible with a translationally invariant connectivity. So these are some of the signatures that come from this, in which the epileptogenic zone, the dynamics within the epileptogenic zone. Yeah. Just for your interest, if you ask mechanistic questions, yeah, for instance, can we go to much more detail? The answer is yes, actually. Within the platform, certain activities needed to be developed to have actually representations on the microscopic level. So there you have to use a simulator which is called NEST, a cellular simulator, and here the virtual brain. If you want them to cooperate, to speak to each other, they speak a different language. Mean fields, so these are point neurons with action potential firings. Yeah, this has become now possible, and what you can do, for instance, like the CA1 region, you can take the hippocampus and represent it in NEST, and then embed it in a simulation using the virtual brain. And the data for this you have available. So this coming from Javier de Felipe's lab in Madrid, where he has essentially scanned the human hippocampus, human, not rodent, yeah, human hippocampus extracted after surgery, and reconstructed all positions in three-dimensional physical space of the cells that were possible to obtain, and reconstructed the arborizations of the dendrites, and the axonal tree. Represented this numerically, and then computed the overlap with a technique called probability clouds, where you actually are able to capture the same statistics as the individual neurons, make synaptic contacts, and build and reconstruct the same region in 3D of the human hippocampus. And now you can simulate it either without TVB or with TVB. And this is actually important because context matters. Yeah. On the left-hand side, without TVB isolated, and here with TVB, and you see that the dynamic changes significantly. And here you can now study questions about how we can actually stop the propagation patterns. Stimulation approaches are being developed in order to do this. Pretty much like we do it in the heart also, where you need to know how to time a stimulus in order to stop a seizure. Today, we do not use this in the brain. Yeah? So this is becoming possible now. So this was kind of an overview of what I want to show you is what can be done in principle in this platform going vertically across the different scales, what data are available. In the remaining time now, I want to go more horizontally and show you how we can use this type of technology now to put it to use in clinical applications. And the framework is the following. Again, digital twin framework. On the left-hand side, real-world representation. On the right-hand side, your virtual brain twin, essentially your mathematical representation of the totality of the knowledge you have. You just formalize it mathematically. Let's call it a digital twin. Yeah? Then you fuse the data coming from the human brain and start actually putting constraints on the parameters, yeah, that you have in the model. How do you do this? You do this typically through inference. Yeah? Not optimization, not fitting, but through inference, because we have an enormous variability. Yeah? So Monte Carlo techniques, typically, most of them that we use are some variation of the Monte Carlo technique. Yeah? And in order to obtain posterior probability distributions, estimating the data, and estimating the parameter you have in the model. And the model needs to be adapted to the type of question you will ask. Yeah. So which means you have to know, you have to have a very good idea about the pathophysiology. In epilepsy, it's the epileptogenic zone. We speak a little more about this when we go in the application. Yeah. In Alzheimer's disease, we discussed that yesterday. Yeah. In the limbic system, yeah, it will be the plaques, the deposits that change some of the parameters. Yeah. In multiple sclerosis, it's the time delays. You have a demyelination of the fibers. Yeah. However, what you find is actually it's called the radiological paradox. There is a very, very bad correlation with the lesion load you have in multiple sclerosis, yeah, and the clinical symptom. Why is that? Yeah, and this can actually be very nicely explained. Pierpaolo in my lab is working on this. It's the time delays, yeah, that enter in there and play a very important role. So you have different representations in the math, yeah, that can be estimated, and this is then the inference process here. Yeah. And what I want to do in the remaining time is I want to go a little deeper in the epilepsy application with you. For epilepsy, what you need to recognize is 1% of the human population has epilepsy, number one. 2/3 of this 1% we can actually control via medication. The remaining 1/3 we cannot control. Yeah, or for them, it's actually, there are two approaches. Stimulation is upcoming, not working too well at the moment, but it's in development. The other one is resective surgery. For this, you need to know what to resect. Yeah? And this is being helped. Typically, you start with EEG. Yeah, you look for lesions in the brain. Yeah, MRI, that's one of the best predictors we have at the moment. You implant electrodes. Based on this information that you have to make a decision of where to implant the electrodes. These are the so-called Talarac needles. They're about that long needle. You have a spacing of a few millimeters contacts. You'll have about 10, 15, or 20 contacts in there that you implant. And then you have this type of measurements here. And based on this, you have to essentially make a decision of where to make the resection. This work from Maxime Beau in Bern, I believe, yeah. Just to give you an idea that over the past 30 years, he has data for the past 30 years, but it is actually valid for the past 50 years, independent of the type of epilepsy, temporal lobe, extra temporal lobe, with lesion, no lesion. There's been hardly any improvement, yeah, in terms of surgery success rate. For temporal lobe epilepsy, we had around 70% surgery success rate, and it goes all the way down to 25% surgery success rate in the frontal lobe. Yeah, so this is actually disastrous. So, and it's being thought that this is due to the bad identification of the epileptogenic zone. That is the key claim, actually. Yeah? So this translates now for us, working in the modeling domain, to how can we better identify the epileptogenic zone? Very crystal clear statement. Huh? And essentially, we are taking this digital twin loop, virtual brain here on the right-hand side, input data, in vivo data that we nowadays we put it into an ex vivo high resolution framework, and we use this data as a prior in an inference framework. So it has to be an inference, so we can actually put priors on this, the individual data, in order to have an individualized prediction for this particular patient. Yeah. So that's what I want to show you. So what we need to make decisions about is what is the data features that we're looking at. Yeah, we talked about functional connectivity yesterday, but these things have never been very predictive. Yeah, for seizures, we're interested in the seizure propagation pattern. Here you have a seizure, brachial spike, seizure onset, very classic pattern, very low amplitude, fast discharges, zzz, becoming louder, and then stop. This translates into, when you look at the frequency time representation, it's the same seizure, brachial spikes, high frequency, amplitude going down, developing harmonics, because you get the spike wave complex towards the end, and then it stops. Yeah? What we are using in our framework is the envelope function, yeah, for every single electrode. Yeah, and we do this on this SEG level, and then we can compute these envelopes. This object, mathematically, if I span it in state space, corresponds to this geometry. This is a resting state, seizure onset, bifurcation, jumps over, spiral, evolves, seizure offset, goes back to resting state. Yeah? This is the object that we want to sample using the MCMC, so the Monte Carlo Markov chain techniques that we're using. And if we use the data feature of the envelope function, what we do technically, we project into this plane here. And this is what it looks like, essentially. So the sampling happens here. We could also go into the individual cycles, but that has not provided any additional information, simply because it doesn't matter, apparently, the phasing of the cycles. Yeah, this has been known actually quite well in the literature. So this is a data feature that we have. And out of this, sampling and the inference process is essentially you generate many different instantiations of your generative model, and you confront it with the data, and using certain criteria, you estimate your probability distribution of the parameters that you're after. So it's not an optimization. This is what you get in blue. It's real data, by the way, now. Excitability for one brain region. If you take a second order approach of very naive variational inference approaches, very often you find this in especially in neuroscience. DCM does it, for instance. Yeah, does a Gaussian approximation. This is what you get here. Yeah. So it's helpful because it gives you a first shot, but you do not capture the details of the multimodality. Yeah. This here means epileptogenic, cut. This means not epileptogenic, do not cut. Yeah? You want to know that. And when you do it not only for a single region, but unfold it in multiple regions, you have a multidimensional parameter space, and you want to sample this information essentially. This information means it's your posterior probabilities. These are the different possibilities with a probability mass underneath, under each spike. Yeah? So this can be formalized and communicated to the clinician. This is what we do. And then we provide a heat map together with the probability distributions to the clinician, and a heat map for the maximum in this for a particular realization you find represented here, overlaid with a posterior anatomical MRI after surgery. Yeah, so posterior, so after the surgery. So in this case, you have a very good correspondence. Yeah? What I want to show you is maybe a use case that is coming out of our lab, a patient that we had. Female has been epilepsy, drug-resistant epilepsy since very, very young age, starts typically early. Yeah, and the clinical symptoms have been known. Usually you go through a cycle of 3-4 medications until you're being declared drug-resistant. EEG, no spikes have been observed. Lesions in 3T, nothing has been found. Going to 7T, 7 Tesla, something in the left orbitofrontal cortex is indicating a discharge, so a lesion, but it was not entirely sure. Yeah, and she was implanted and explored with SEEG. This is the area of the lesion that may be visible in the 7T. You see some of the discharges and also the pattern here. The delayed onset, you see the fast discharges here, some tingling, and then the fast emergence until it slows down at the end. Yeah. So this is the corresponding SEEG. Now there are certain procedures we talked about inverse solutions yesterday, didn't we? Yeah. So you can actually, there are different techniques, high resolution EEG. Loretta, some of you may know low resolution tomography can actually map on the cortical surface. It provides you with this. The FCD, the dysplasia is located here, no correspondence. Yeah. Same situation here. Simultaneous EEG and MEG has not necessarily improved the localization.
MEG does not?
What?
MEG does not?
It depends. In this case, this is a clinical use case now. In this case, not necessarily. MEG helps, and we do this regularly. We have an MEG actually in-house on the clinical sites. So many of our patients go through MEG. Yeah. Here in this case, it did not help. When you analyze the data, so I'm in full support of MEG, in particular with the OPs, the novel technologies to actually optically obtain these particular signals. Wonderful, very promising. Yeah. Here, just to give you an idea, different indices can be computed on the SEG data based on connectivity. They have indicated into two regions, left orbitofrontal cortex and left insular gyri. Yeah. So when we performed with the workflow that I showed you, the estimation of the heat map, we got a pretty good correspondence plus one additional area. Yeah. There certainly was not as large, but 3 epileptogenic zones that were identified in there. Patient went through surgery. Yeah. They have decided to remove the first 2 areas, yeah, which is a correspondence with VEP of 67%. Yeah. After 1 month, she had seizures again, yeah, and she was classified angle 3. There is a classification scheme. Angle class 1 means seizure-free. Angle 2, improvement, but some seizures. Angle 3 is essentially not good. Yeah. So what we did then, the clinicians came back to us and asked, what is about this additional area that you have identified? Then we went back into the MEG and zoomed actually into this area that was identified by VEP, virtual epileptic patient. This is how we call this workflow, and actually identified spikes, intra-ichthal spikes in this area. After further, so that was another indicator, and together the evidence indicated we may go for another surgery. She went through surgery last year in summer and is still seizure-free since then. Yeah? So this is one of these success stories where it was actually part of the clinical routine, this process. You can go now retrospectively, and this is a little bit what we discussed yesterday, and compare the performance for 54 patients retrospectively and say, how does the heat map overlay actually with the clinical hypothesis? Yeah, this is precision. There are different statistical metrics you can use. In this case here, it's precision. The precision is pretty good, 77%. The patients that is seizure-free. Jumps down for not seizure-free patients. Yes, so seizure-free, not seizure-free. And if we compare it against post-surgical MRI, what I showed you earlier, yeah, then this is the clinical hypothesis, precision. This VEP, very good correspondence, but here it shoots down completely. Yeah, and this and other indications actually told us that there is probably some added value in performing the VEP because of this strong correlation in the retrospective data of surgery failure and not correspondence to what came out of the prediction. And this is even improved when we go now to high resolution, from low resolution to high resolution. I want to show you this one here in particular. Look at the precision value, seizure-free. Look for high resolution, how it starts nicely gathering up at very high precision values. And that's due to this improved source to sensor mapping, which makes perfect sense, but it's nice to see in the patients. We had only 14 patients here because out of the 25 patients, the 11 had too much atrophies in the data. We were not able to reconstruct the high resolution representation. This is a disadvantage here. So what, in summary, what is in there? What is happening there? Where is this? Is it simply better identifying what the clinicians are not seeing, or what is happening there? Yeah, this is what I want to illustrate you here. You do EEG for a particular patient, MRI, PET, whatever you can, preclinically, and then you make the decision about an implantation, SEG implantation. This is the first big invasive step. Afterwards, based on this, you make a decision about the epileptogenic zone. Yeah, and typically from there, you make a decision to do surgery or not. What we are doing, we are adding a full brain network, not just here. It's based on structural data. not on functional data. It's based on structural data. And then we perform partial sampling. We essentially inform the whole brain network, only here, but through the maps from the sources to the sensors. And here is essentially the connectivity that enters a particular organization that we have in terms of this. But we cover with a model the entire network, which means we can also make predictions about this, the so-called missing electrode problem in epileptology. There may be areas that are outside of the accessible area for the electrodes, but we capture it with that, yeah? In principle, in theory. Does it work? And the answer is actually yes. This false discovery rate for the patients I showed you, 25 patients, clinical hypothesis, seizure-free, not seizure-free, same, yeah? For BP, very small value, false discovery rate is good. Yeah, and it shoots up for those that are not seizure-free. And 29%, a third of all the regions that are corresponding to the surgical failures, they fall into the category that are outside of the accessible areas for the SEEG. And I'm answering your question. What is the added value of a model? Yeah, medicine. It opens up the range that is invisible to you. Yeah, that's another possible answer that one could give to you that you asked yesterday. Yeah, where are we? So that's what I want to share with you. Where we're going with this.
In how many patients do you know if they have a protection like
Yes, 54. 54 patients. And what we are doing now, we're doing this prospectively. Since 2019, we are running a clinical trial in France with 13 reference centers, yeah, all over France, independent centers that are collecting data, structural, functional, SEG, following this routine, centralizing at Lyon. We are accessing it. We are virtualizing the patient, 400 patients, yeah? Or almost 400, and performing, passing them through the pipeline and providing them with a report with a heat map and sending it back to these centers. And there are essentially two branches. One branch, half of them receive the information, and the other half not. We call this VP positive and VP negative. And the clinical trial has just finished in June this year, at least patient inclusion finished. Yeah. So we had 356 patients. Yeah. So 300 patients randomized, some of them got excluded, 178 surgeries performed following this. Yeah. We have to wait for 1 more year because not all surgeries have been performed. And we want to wait 1 year until after surgery to be able to judge actually if the patient is indeed seizure-free or not, because this is our criterion. Primary objective is, can we improve the surgery success rate? I remind you, the one that has not changed for the past 50 years. Yeah? And the second objective is to learn better actually about the intricacies of the technologies, the modeling, the inference, etc. That's what I want to share with you and show you. Essentially, this is the workflow that we hope for, translating it into clinical applications, in vivo data, merging it into this framework, and then performing this inference. So it has to be inference, not just optimize. You need to know how much you know or how much you don't know, actually. That's why it's important to have an inference framework with posterior probability distributions. If you don't know, you want to know that you don't know. This is very important. That's why we chose this admittedly complicated approach. This is a vision for the future, and we are working on this. I wish to thank my colleagues, my friends that have worked to this, and also the funders. Thank you. Yeah.
So thank you very much, Viktor, for this beautiful presentation. So, and that opened the for question.
Okay.
Thanks
But
a lot.
This figure up, I like it. Yeah.
Yeah.
So I like playing with numbers.
Thanks a lot, Victor. It is an amazing talk, really impressive work, impressive results. I have really a lot of questions. I will only do the main ones. Just to clarification, and it's a follow-up from what I asked yesterday. So here you were saying at the input, so of course you provide data from the anatomy, from diffusion data and so on. And then from SEEG point of view, you said you mainly introduce the envelope of the signal.
We could do something else, but we choose to do the envelope because of the technique that we use. Sorry.
So my first question is, how many or what is the influence? Like, are you putting into the model one seizure, several seizure, as much seizure as possible? That would be my first question. And my second question is what, following a little bit Shifa's comment, what would be, what is happening? Because you know, all implantations are not, some are very well done, and sometimes we are just missing the main focus, and we know more or less that we are getting mainly the propagations. So do you have an idea of how the model is behaving in those cases? Like basically, these are two different things. These are you catch the focus, but there's a second one, which are the red zone that you put, or you missed the focus. In that sense, your implantation was not well done. And how the model is behaving, because the data you put is not the ideal data that you put as an input in this second case.
A little background for the last question. When you have the SEEG electrode, each contact samples approximately 1 centimeter around it,
maximum.
not more. Yeah, so it's so easy not to sample any other epileptogenic zone. That's why the missing electrode problem is so important. Let's start with the first question. Best thing is I tell you how the clinical trial protocol is organized. We get typically 1 to 3 seizures. Spontaneous seizures are preferred. If there are no spontaneous seizures, we work with stimulated seizures. Yeah? This is a number of seizures that we typically have. It is sufficient to perform a reconstruction. At the moment, we use as a data feature the convolution, the envelope function. Yeah, that was a choice because we use a Monte Carlo technique. Today, so 4 years later, we would do it differently. The techniques in machine learning AI have evolved enormously in the last 4 or 5 years. There are techniques such as simulate-based inference, SBI, for instance, which is superb, which is what we use today, but this is in the research domain, not in the clinical trial. Yeah? And for the second one, for the second question, we as you see from the data I showed you, 30% is estimated epileptogenic zones outside of the accessible zone. So that tells you already that there is some predictive power with regard to this. In the low resolution virtual brain framework, shockingly, we have discovered only very recently in the last half year that there is a higher sensitivity to the location of the SEEG contact. Moving it by a few millimeters will actually not change the results completely, but the organization of the ranking of the elements. That is due to some reasons. One of the reasons is a very bad mapping from source to sensor. Imagine the following. If you have the 16 square centimeters and you fold it something like this, if you have high resolution, you have also the dipolar momentum. Yeah, that if you reduce everything to a single point, you have no momentum, so you're taking only the distance information into account. That's bad. Yeah, not good enough. That's the key reason why we had to go to a high resolution. This improves it enormously. But the clinical trial started 2019. Again, what would I do differently today? But this is the way to go. So we find on the low resolution large sensitivity to this. This is becoming much better in the high resolution. We find many regions outside of the accessible region, and we find evidence that this is predictive. Yeah. Beyond that, I cannot tell you yet anything. But I will show some patient data in the other talk. Yeah. Yes.
I have a naive question. My question is it possible to build a model without using ECG data from the patients as a prior or not?
Yes.
So
We don't need SEG data. Yeah. The model building happens entirely based on structural data, ex vivo or in vivo. And then we put in the appropriate mathematics with which we work. Where the personalization comes in, that we need functional data. The first level of personalization is you use the individual's own structural data. This is, of course, a level of personalization. Second is you do the second is essentially functional data. Can be SEG data, it can be fMRI data, it can be EEG, MEG data, something that is actually fantastic. Are you an epileptologist?
yes.
Yeah. Fantastic in this workflow. Yeah. We can demonstrate the following, unpublished, but we have demonstrated now proof of concept. Yeah. We can do the same thing with EEG. Why can we generally not use scalp EEG data? Because we don't have any seizures. Ictal spikes, you see them sometimes you don't, etc. Yeah. There are new technologies upcoming, yeah, in particular for stimulation. I'm not talking about TDCS or something like this. TDCS is just close to the surface. But there are new techniques such as temporal interference that allow, yeah, that allow you to go profound. In our house, we have Adam Williamson that uses actually with multiple nodes, allowing to take the focus and reduce it to a size of 2-3 millimeters. So suddenly you have a deep focus of 2-3 millimeters where you can stimulate. We have demonstrated proof of concept that we can do this non-invasively, trigger a seizure or not. So we can do the sampling as you do it in the clinics with your patients when you go through a grid, stimulate one electrode, change the frequency, etc. If you're interested, I can show this to you. We can do the same thing in a patient-specific manner, and we can actually replace this workflow with an invasive SEEG by EEG, non-invasive, trigger the seizure, and based on the data, we can actually perform a reconstruction. Is it better than SEEG? I don't know, because sometimes you have a missing electrode. In EEG, you have no missing electrodes, but you have all the issues that are associated with the scalp EEG. So we need to run studies on this, but we have proof of concept that we can render this type of preclinical exploration non-invasive in principle. Very early times, but very exciting.
Thank you.
So you never use fMRI?
Yes, we do,
You do?
but not in this example of EEG. We use fMRI for, you will not hear, Randy spoke about this, for in aging studies. For instance, we have built an entire healthy aging cohort. In Alzheimer, we use fMRI data, etc. fMRI data are, it's a different domain. The same principles apply. That's why I showed you this translation to the clinic. You can paraphrase every single question depending on the pathology and the corresponding pathophysiology and the imaging technique. And every time you have to ask the same question. What is the data feature? Is it informative? How do I represent it in the model? I go through the inference cycle, yeah? Is the pathophysiology that corresponds to that identifiable or non-identifiable? What we have shown, for instance, in dementia, yeah, that in a resting state fMRI, we are the plaques, if we want to detect them, we don't manage to do this in the resting state paradigm, but we manage to do it in the stimulation paradigm. If we ever manage to get this temporal interference set up into an MRI and then start sampling the individual brain areas and then perturbing, and then they get back, yeah, this relaxation samples much more information and renders actually the pathophysiology of Alzheimer in the context of the model identifiable, which is pretty cool. Yeah.
Yes.
Very beautiful presentation. Very nice. Thanks. So my question is the following. Let me do an example. Okay. So you have a car right? Then you
I-
know to you have. Good. I will not ask which model you have. You have a car, and and you get a flat tire.
My
Okay?
wife does.
Yes. So
Regularly. Sorry.
so you know to model a car, right? Now, let's say that one of the parameters for modeling the car is the radius of your tires. Okay. Now you get the flat tire. So then you know that there is something wrong with your car, right? You will experience. And so one thing that you can do is say, okay, I know I have my model of the car, right? The healthy car. And what I do is I try to fit the pathological situation. And so what I can do is just reduce the radius of one of the tire, right? And so then I can see if with reducing the radius of the tire, I can fit the behavior, right? I can model the behavior of a car with a flat tire.
Mm-hmm.
But we know that that's not enough, right? A car with a flat tire is not the same as a car which has one of the tire with smaller radius, right? So now what we usually do, and what I see you are doing, is exactly taking the model of a healthy car and change according to the pathologies one or the other parameter. You say, let's say, problems with myelination, then you change the tau parameter, the delay, right, in the transmission or whatever. So, but it's always the same. It's starting from a healthy model and adapting the parameter of a healthy model to describe a pathological situation. But my point is there any effort, right, in the virtual brain and your approach to start modeling directly a pathological condition without starting from a healthy situation and just changing the parameter in the healthy model?
Wouldn't you agree you always start from the healthy model, and then I think you're talking more about macro and micro representation, because what would be an example of modeling the pathophysiology? In multiple sclerosis, the example that you just brought, it's the demyelination of the fibers. Yeah? So you see actually the callosal volume is going down. This is modeling the pathophysiology.
Yes, what I'm saying is that the point is that sometimes, right, a pathological behavior complicates the situation. And by just changing a parameter in a model that was working for a healthy situation may not be enough, right? So if you want to model a flat tire, you cannot just use the model of a healthy car and just change the radius of one of the tire to pretend that you are modeling a pathological situation.
What if I change the elasticity properties of the tire also? Would you be then happy?
Yeah, so you need to do more than that.
Yes,
right? That's what
so-
I'm saying.
So not just one parameter, but multiple parameters, or potentially also go more microscopic in order to have more biophysical detail in there.
Yeah.
Yes, this is multi-scale modeling, and we do this. In the beginning, I showed you the co-simulation of NEST and the virtual brain, that we are capable of doing some of these things. I'm not driving this myself. Yeah, this is more our Italian friends such as Michele Migliore, etc. I typically stay on this macroscopic level. However, I want to know how microscopic features actually inform my macroscopic parameters. So in this sense, we are doing it, the multi-scale cascade of cause and effect, which then provides multiple parameters, if they matter or not. This is being done typically separately. That's one of the approaches that we are working on, where we are modeling this typically in a high-dimensional, more cellular approach, and trying to sample the different types of behaviors. And then we are, bless you, and then we are taking the outcome, typically as parameter manifolds, and then use it to constrain our macroscopic virtual patient model. It's a little complicated. One naive answer would be, I can build a cellular model of the brain and represent every single cell, etc. This doesn't work. You run into the parametric explosion. Other people have tried that. You get completely lost. Yeah. So you need to use smarter approaches. Yeah. So what we are doing in our hands, we estimate the cause effect from microscopic to macroscopic. This is all ongoing work at the moment. Yeah. And this cause effect, we try to parameterize it. Yeah, pretty much as what we discussed yesterday when you gave your when you gave your presentation, multiple qualitative behaviors as a function of parameters, and then we take this behavior and implant it into the virtual brain model, assuming there is an assumption behind there. We call this weak coupling in nonlinear dynamics, assuming that the basic characteristics of the network node of the dynamic system do not change when you put it in a coupled network situation. This is a strong assumption. All of us do it all the time, whether you say it explicitly or not. Yeah.
Okay. Yes.
If there are no other questions, I think my question yesterday may have offended you, and I've just sent you a message, but I want to apologize to all mathematicians in this room that I absolutely have no doubt that mathematics is the basis of every single knowledge and advance that we have made in medicine.
Not at all. I apologize to you for him. No, what I meant to say is afterwards, we discussed actually, Randy and I, what is the best way of responding to you? So we were reflecting about this. And I said to Randy, I will mention it tomorrow.
No, absolutely. So,
no
I'm
offense
a biomedical engineer, so I have played with mathematics. Yes, so now all I wanted to say, I think I was referring to the issue of using mathematics to address things such as psychosocial determinants of health.
That's what he said.
So there, I think we might have a bit of difficulty with mathematics, but in medicine, we can't do it without it.
No, you placed it very nicely into this social context, and Randy responded to this very well. Yeah.
So,
But it's a question that can go wider. Yeah. So it's a very important question. So.
Hello. Thank you for a great talk. I'm more research on embodied cognition, working virtual reality and all these things. So mathematics and neuroscience, I just have a taste of everything. So might be a naive question, but when I looked at the folder, what I was very interested to see was this embodied cognition actually coming back into this pipeline, the simulation environments. And so my question to you, and it's like say maybe a naive and broad or high level question, but I'm curious about your answer anyway. I'm thinking of when I see this modeling of the brain, of a brain in a vat. And as I'm sure that many of you might follow, is that we are not just brains in a vat, we are interacting with our environments and so on. So the robotics thing. So I'm curious, and also for the future, with these more simulated environments, virtual reality, robotics, like you say, prosthesis, but also for your clinical work, we're not just brains in the vets, interaction with environments. What's your stance or strategy of maybe placing them back into the real world, or creating more richer context to maybe inform the modeling itself?
Yeah. Very good question. Very important. I myself am not working on this in this domain, but here I'm responding as the Chief Science Officer of EBRAINS. We are very aware of this, and world modeling. So external world modeling plays a very important role. People such as Egidio D'Angelo, for instance, or Rainer Goebel, for instance, they are working on this. What Egidio does, what you find in eBrains, it's by far not as far advanced as we'd like to have to. Yeah, there is a lot of activity in software development for external world modeling. So we don't want to recreate it, but what we created is a so-called NRP, Neuro Robotics Platform. The name is a misnomer, it gives you the wrong impression. What it does is it allows you actually to plug different softwares together in the same environment. You essentially put the software, you unpack it in its own, so it's a virtual box, you unpack it, and then it can operate interoperably with the other softwares. This is what we're using at the moment, which allows us to use different types of softwares. There you will find a representation of a rodent skeleton that you can, for instance, Michele and Egidio do this, use a cognitive architecture, coming from the rodent, you can actually use in order to control muscle fibers and have the rodent operate on a treadmill. If you don't want to have it that fancy, you can reduce it to a point and then navigate in a spatial environment and interact with visual signals, move, locomote, and you have obstacles that you can work around there, all using the cognitive architecture, not like connectome-based, but much reduced, modeling individual areas. However, it's this representation. This is actually where it's coming from. You have multi-region models that are represented by spiking neurons using nest on neuron simulators, but classic cognitive architectures. So predictive coding, you have an internal model and you want to navigate in this space, or spatial navigation was what was used mostly there. So this is possible, by far not as much as we want to, but there are one or two use cases that may be of interest to you, and the technologies are in place, but we need good users that build better use cases. So it's really the beginning.
I saw this Angora Pi also, so I made a note of that, and I will definitely look it up.
Angora Pi, Rhino Goebel, exactly. That's, you made a very good note.
And a small follow-up, but if I can, last one, something about timing is important. About social cognition, is there anything more social in the whole brain simulation project? I mean, we're talked about this.
In terms of data modeling, nothing. In terms of data, yes. For instance, the data sets, the 1000 brains cohort that has been collected in Jülich, healthy aging cohort from 55 to 90 years, comes with beautiful structural data, functional data, all MRI, fMRI, but also it's been calibrated very beautifully with cognitive tests, filled out questionnaires, you know, if they are doing physical exercise, reaction tests, etc. All this data, all this information is available. On this level, yes, rich data sets. In terms of modeling, very poor, nothing. I cannot think of another dimension at the moment.
Okay, thank you.
Okay, thank you. Comment and question. Comment is about EBRAINS. You mentioned that we can store, the storage is for free. So
for a standard user.
Standard user, not the medical
if you
data.
then start for a standard single user, and within reason. If you then want to go beyond that, reach out and interact with us, and we find solutions. So it is for free for standard research use.
Okay,
which makes sense.
This is clear. It makes sense. Yes. And have you solved the problem of regulation of the clinical data?
Solved. It's going to accompany us for many years, but what we have today, and it is functional. There are three approaches. I'll give you the quick answer. One is called Health Data Cloud. So there is a cloud system with multiple layers of controlled access. So this is the way how to do this. This is actually Petra's work, Petra Ritter. So you have cloud access, different layers of controlled access. This is one possibility of doing it. Second one is a HIP, human intracerebral platform, data platform. This is specific for SEG data, effectively, and protected environment in Geneva. Also controlled access. So that is different organization. Very specific, but it is functional as of today. If you get an account, you cross all the barriers for authorization, you can actually access some of the SEG data today. Well, Your account, if you apply for your account, it will be approved within 24 hours. Yeah, this is a promise. Yeah, and then you can get access to the different users. Number 3, MIP, Medical Informatics platform. There, I think it's about 40 hospitals that participate. You go behind the firewall of the hospitals, work on so on the hospital servers are provided, and you put we refer to this sometimes as EBRAINS in a box. Yeah, you unpack it on the server, and your functionality is very limited. However, it allows you to access patient data. No patient data will ever leave the hospital, but some of the results can leave, and then you can process the appropriate results statistically. Good for rare diseases. Imagine each hospital has 2-3 individual cases times 40. Suddenly you have a cohort where you can get statistical significance in a way where you are never ever be able to do it in any differently, yeah, because simply the patients do not exist.
Okay.
So different use cases, different scenarios.
Okay.
Have we solved it? No.
No.
We're working, but it's getting there.
Absolutely. So this is a general problem that we
Yeah.
all have. One question. Oh, you answered this question about the multi-scale approach, because you present the NEST approach, but in your for epileptic seizure, you don't use this multi-scale. You are at the macro scale level. So all the features that you're talking about doing prediction are at the macro scale level.
Yeah,
is that right?
For the macro scale level in application to patients in clinical use, we are completely at the macro level. For research applications, we are using the co-simulation, and we are working on the spiking network.
Oh, okay. Well, so my question is, when you connect this micro scale level, is there any, I mean, adding value to the prediction that you are looking at?
So far, not in our hands.
No.
Yeah.
Okay.
So in our hands, so far, no.
Okay.
Yeah.
Okay. Last question is about the.
But it's insight,
it's
mechanistic insight that we
I
generate.
expect showing something coming from this multi-scale modeling and predicting this.
And something may come out of this. So far, not. Yeah. It's starting right now, going to the high resolution modeling. For instance, like stimulation, usually you don't stimulate as a function of the traveling wave at the high resolution. We can actually show, and in cardiology, you do that actually. You measure the propagating waves on the heart muscle, and then you stimulate as a function of the phase. In epilepsy, not at all. Now here at high resolution, for the first time, we can even envision something like this. Until now, it was not possible to even formulate the question. So it is possible, but we have not done it yet.
Okay. The last part is about the prediction. So using the Bayesian framework. Bayesian framework, simulation, back to the data, and doing some prediction and evaluation. So the prediction and the distribution that you show are the features that you give to the clinical side, and they decide based on this kind of prediction. So it's correct, incorrect.
Yes.
So
you have a staff meeting where the responsible clinician presents a patient, and typically the patient is that age. This is a clinical history. MRI negative, MRI positive, EEG has shown the following, etc. So you go through all these elements, and VP becomes one of them.
Okay, and the role of the virtual brain, which is the simulation side, it's mostly to simulate the activity of the different areas of the brain, and the feature that you are looking at or you extract from the data is the envelope of the SEEG or the envelope of the LFP that you simulate. Is that right?
No. The data feature of the empirical SEEG, this is a data feature that goes into the inference framework. Careful, it's a causal inference framework, which means we have a generative model. This is a virtual brain. That's what we are sampling. The simulation itself, I have to answer carefully. The simulation itself can either generate the full seizure at the highest resolution,
Yes.
or we also have a reduced version which simulates only the envelope function. So yes and no, both variants exist.
Okay, so my understanding is that the simulation comes with this envelope or the whole dynamic of the system.
Yes.
And the inference goes to the estimating some parameters to do the prediction. So my question is about the personalized parameters that you change, you turn in the simulation and you estimate using the Bayesian approach. What is your experience on that? Depending on, do you almost the time estimate the, I would say, the right parameters? Because the Bayesian inference needs some a priori, and those a priori may change depending on the context and how this affect the estimation of the parameters and the prediction that you are looking at. Or maybe it's enough strong using like non-informative a priori, and it's enough to do a kind of prediction. What is your experience?
We've done a lot of work on this. The last 5 years, we have done essentially nothing else. Well, not nothing else, but put loads of emphasis on this. The most detailed simulation you see here. It's not just envelope functions, it's detailed discharges, etc. The a priori typically is if we have a lesion, for instance, if the patient is MRI positive, then we actually narrow the prior in certain areas and we bias it a little towards the lesion. Otherwise, the prior is actually quite wide because we want the sampler allow to explore different alternative and different solutions. How well does it infer the epileptogenic zone? It depends. It depends on the patient. Sometimes patients have different types of seizures. Yeah. Sometimes a epileptogenic zone is not identifiable. We can get always results if we bias it with the priors strongly, but that doesn't mean that the posterior this typically means that the sampler got stuck at the prior and was not able to find other solutions. Sorry, this is becoming now a little technical, but essentially it's a sampling paradigm and it's stuck at the prior. So we need to allow it to sample other possibilities. It can absolutely happen that the posterior probability distribution is quite wide, not uniform, but wide, and we cannot make a very knowledgeable statement. And this we pass on to the clinicians. Essentially, we are not sure. Yeah. The multimodality, you saw that is reality, which is actually very, very cool because you with your patients see this also. This is reality. Yeah. Sometimes you cannot say, but you can actually say whether this is a good candidate for the zone, epileptogenic zone or not. There are several possibilities. So we see everything you can imagine: multimodality, non-identifiability, very strong convergence. It depends on the seizures of the patients, on the implantation scheme, and etc. So we are very careful with the sampling, and if it doesn't converge, we have multiple criteria and quality checks, which we have to be in a trial, yeah, to make sure it converges. But we also know when we don't know, and it happens more often than we want to.
That's great. Okay. So thank you for this response. We are going to stop this discussion about this first part of the